It's been a long road home, a long road to Homo sapiens for Neanderthal, who was discovered over 100 years ago in the late 19th century and portrayed for several generations thereafter as a missing link proving evolution. Originally, they portrayed him as an ape-like creature covered with hair, with no clothing at all, a broad-based stance, and hunched over as though he's trying to evolve into bipedal locomotion. Yes, he's carrying a rock like a chimp might, but he has a stick in his hand. So they're saying that maybe he's evolving tool use. But it eventually became known that Neanderthals were buried with clothing, and so they upgraded him somewhat and moved him to fully human by putting clothes on him. But they still want you to think he's kind of dumb and evolving. In this picture, we see that the Neanderthals have invented clothing, although they haven't invented hats yet. And they're burying their dead, something that chimps don't do, apes don't do that, and so he's fully human, and the burial is in a ritualistic posture with the knees drawn up and the head turned towards the right shoulder, the body covered with ochre. All these are correct according to the burials they've found with Neanderthals. Notice that the men are using rocks to shovel the dirt in. They haven't learned how to haft sticks onto rocks yet. And the Neanderthals are hunched over as though they're still trying to evolve fully upright posture. Now, this diorama was fraudulent from the get-go. They have no clothing, Neanderthals here. They're completely naked and have no tools in their hands. But this was known at the time to be fraudulent. A better diorama is pictured here, and they've upgraded Neanderthal in this one. Notice that they're wearing clothes. They haven't invented hats yet, although perhaps this fellow down here has a hat. Notice that they have no stone points hafted onto their sticks, although they are using fire. This structure is a poor excuse for a pit house, and the Neanderthals here are still hunched over with a broad-based stance as though they're having trouble evolving to fully upright posture. And they want you to believe, the evolutionists want you to believe, that these men could not speak, but they grunted like apes. Into this controversy comes ancient DNA, which this is a study published by the David Reich lab in Harvard in 2014. And what they did was they had a fully sequenced Neanderthal genome, and using an advanced statistical method, they found motifs in the Neanderthal genome, which they searched for in modern human genomes. And lo and behold, they found Neanderthal DNA was everywhere. Everyone seemed to have some Neanderthal DNA. The least amount is in Africans, but some groups have up to 1.5%, and some individuals even more. Now, this is not a small amount of DNA. When you consider that 1.5% theoretically could come from a Neanderthal ancestor who lived 6 generations ago, because you have 50% of a parent's DNA, 25% of a grandparent's DNA, 12.5% of a great-grandparent's DNA, and by the time you get back to 6 generations, you have 1.5%. And so you could have had a Neanderthal ancestor at the time of George Washington. Now, of course, that's not the reason for this. The reason there's so much is that it's widespread in the human race. So you probably didn't have a Neanderthal ancestor at the time of George Washington. Neanderthal skull features are present in the world today. The man on the left is Nikolai Valuev, a heavyweight boxing champion from Russia, and he has Neanderthal skull features, including a heavy brow ridge, sloping back forehead, elongated skull, occipital knob, and wide nasal passage. The chin, however, is a modern chin, since Neanderthal's chin sloped back and did not poke out. The man in the center is from Papua New Guinea, and face on it's hard to tell, but he has Neanderthal skull features with a heavy brow ridge, sloping back forehead, and wide nasal passage. Into this controversy comes another study from the Max Planck Institute in Germany, and this is Y chromosome sequences from Neanderthal and Denisovans compared to modern sequences. And this paper was published in 2020, and it's by Martin Peter et al., and I'm going to use those terms Petter and Max Planck data interchangeably as we go through this. And basically, before we describe this paper, I need to define Denisovan. Some of you may not be familiar with this group. Denisovans are similar to Neanderthals. They were found in the same cave in Siberia, actually. And the first Denisovan specimen was a molar tooth. And when they sequenced DNA from it, they found that it was very similar to Neanderthal DNA, but the mitochondrial chromosome was similar to modern mitochondrial chromosomes. So they put the Denisovans in their own group, but they're similar in their nuclear DNA, and therefore their skull features may be similar to Neanderthals. We haven't found any Denisovan skulls or full skeletons yet, so we don't really know about that. But both Denisovans and Neanderthals were found buried in the same cave in Siberia, although they were buried at different levels. 5% of the DNA in modern Melanesians, Aboriginal Australians, and Filipino Negritos is Denisovan, but the mitochondrial sequences are different from the Neanderthals. Now, this is from the paper, the Max Planck paper, and it shows the location of the specimens used in their study. There was one Neanderthal from the El Cidron cave in northern Spain, a Neanderthal from the Spy cave in Belgium, a Neanderthal from a cave in Russia, and the two Denisovan specimens were from the Denisova cave in Siberia. This is the El Cidron cave in northern Spain, and it's a huge cave. If you look here, that's a man standing there. So this is a very large structure, and you can see why the ancients would like to live there. The cave in Spy cave in Belgium is much smaller. You have to duck to get inside. And this is the Mezmakaya cave in Russia, and it's a huge cave also. Here you'll see a road with some people on it. Now the specimens were as follows. The Mezmakaya specimen was an infant skull fragment. The Spy specimen was a right molar tooth, and the El Cidron specimen was a bone fragment. This is the Denisova cave in Russia, and it is a moderately sized cave. You can see some people here. And the Denisova specimens were both molar teeth. Now, these are thought to be between 55,000 and 106,000 Darwin years old. And I use the term Darwin years here because these were not based upon measurement, but are ideological years. So they sequenced DNA from these specimens. So we need to talk about it. Sequencing of ancient DNA is somewhat controversial because of the difficulties. When they first began to sequence it, they found modern bacterial and modern human DNAs contaminating the specimen. But then they learned to treat the specimen with hypochlorite solution, which removes the intact long-stranded DNAs, leaving behind the fragments. Ancient DNA is highly fragmented, so this cleared the contamination problem. They work in a sterile environment in a hood in the lab, and they drill deep into the specimen to get the sample. The best specimen of all is a petrous bone from the skull, because this is a very thick bone that contains the vestibular apparatus of the ear, the inner ear, and it is loaded with DNA. So if they have this petrous bone, it's a good source. They use only DNA with deamination at the fragment ends, and this is because DNA, when it is decomposed, the cytosines are deaminated to uracils, and so this is a marker of ancient DNA. This deamination can be reversed chemically, and they do that before they do the sequencing on the fragments. And finally, once they get the genome sequenced, they restrict any analysis to transversion polymorphisms. So we need to talk about that. Now here on the right we have the 4 nucleotides. We have 2 purines, adenine and guanine, and 2 pyrimidines, cytosine and thymine. And in a mutation, if one purine is substituted for another, that's a transition. Or if one pyrimidine is substituted for another, that's a transition. And these are indicated by the red arrows on the diagram here on the left, where adenine and guanine are substituting one for the other is a transition, and likewise a transition from cytosine to thymine are back. Now, if a mutation occurs that substitutes a pyrimidine for a purine or a purine for a pyrimidine, this is a transversion, and that's indicated on the diagram on the left by these blue arrows. Now, when we're talking about ancient DNA, we have to consider whether or not the change is due to decay of the molecule rather than an actual mutation that's been inherited. And when you think about decay of the nucleotide, a little bit of decay to an adenine may make it difficult for the sequencing machine to tell if this is an adenine or a guanine. And likewise, a little decay to a thymine may make it difficult for the machine to tell which pyrimidine this is. It takes more decay of the molecule to confuse the machine to substitute a purine for a pyrimidine or vice versa. So the transversion mutations are thought to be more likely inherited mutations rather than simply artifacts of decay. All right, let's look at the data from the Max Planck study. This is a reproduction of their phylogenetic tree for the Y chromosome You use software to make the tree from their data set. And it's oriented with the root of the tree on the left and the leaves of the tree, which are modern sequences or ancient sequences, on the right. So if we look here at the root of the tree, we see that, first off, it's rooted in a chimp ancestor, which means that it is an evolutionary root to this tree. And on the right, we have these sequences. The text in green are modern sequences of the Y chromosome from around the world, about 20 specimens. And also this blue lettering here is the A00 sequence, modern sequence from sub-Saharan Africa. The ancient sequences are all in red, and we see the 3 Neanderthals clustered together here and the 2 Denisovans clustered together here. Now, their tree has all modern sequences descending from a common ancestor who lived in 2500 BC. This would be Y chromosome Adam, or YMRCA. And that man shares common ancestry with a Neanderthal ancestor who lived in 370,000 years ago. And that man shares common ancestry with the Denisovans who lived 700,000 Darwin years ago. So this is the way they've analyzed the tree. Now let's look at the sequences. They are from around the world. This colored map on the right represents the haplogroup sequences of the Y chromosome from around the world. And we see that we have sequences from all over. There's 5 E's and 4 J's and 4 O's, and the rest are 1. Now you might ask, are Shem, Ham, and Japheth represented here? And the answer is yes. According to Jeanson's rooting of the tree, Shem is N, O, Q, R, and S, Ham is A, B, and E, and Japheth is I and J. And there are 5 ancient sequences. Now, the conclusions of the PETR data are these. 1, all modern Y chromosomes have descended from a common ancestor with Neanderthals. 2, Neanderthals descended from a common ancestor with Denisovans. And 3, all 3 late Neanderthal chromosomes, Y chromosomes, cluster together and fall outside of the variation of present-day human Y chromosomes. Now, this number 3 is the main point of the paper. The thing they want you to take home is that Neanderthals differ from modern sequences, and therefore they state clearly that all late Neanderthal chromosomes cluster together outside of modern sequences. Now, I have some problems with this conclusion because PETR's conclusions are a matter of interpretation. First, the tree was rooted in an evolutionary fashion, and this will make everyone descend from Neanderthals and Denisovans. That's an artifact of rooting the tree with the chimp as an outgroup. The other problem is there's only one A haplogroup sample. Now, the problem I have with that is that if there's only one A sample, and then they say that the Neanderthals are outside of that, the A is the closest sequence to the Neanderthals and Denisovans. And anytime you compare things, if you compare something to one thing, it's always going to be separate from that one thing. I mean, I'm separate from the wall here. And if I go halfway to it, I'm still separate. If I go halfway again, I'm still separate. I'm always separate from this wall on my left. But I'm in the midst of these 4 walls. So to make a comparison, you really need multiple specimens to compare to. And so this brings up the possibility that a different interpretation would occur if the root of the tree were made to be Noah and the tree oriented in a young earth creationist fashion, and if more samples in the A haplogroup were added to the tree, one might get an entirely different interpretation. So that's what I decided to do. The method was this. I would add A and B samples from the Carmen data, which is a large Y chromosome data set I have available, and I would restrict the data to the 6.9 million bases of Y chromosome used in the PETR data. I would use only transversion SNPs, and I would root the tree in Noah. So I combined the PETR data and the Carmen data into a VCF file in 6.9 megabases. I made a multi-FASTA file. I used Mega10 software with a neighbor joining algorithm to make a tree, and I visualized the tree with Fig Tree software. Now, the Fig Tree programs and the Mega10 programs are available online to help make these phylogenetic trees. And here's what I got. This is the PETR data combined with A and B haplogroups from the Carmen data and the tree rooted in Noah. Now, the first thing we'll notice, this big old blue arrow here is the Noah root of the tree according to Jeanson's work. and I put that at 2500 BC, or about 4,500 years ago as the time of the flood. The green sequences are the petrodata, and Shem, Ham, and Japheth are represented here. Shem is these sequences O and N and S, Q and R here, and Ham is these A through E sequences here, and Japheth is these I and J sequences here. Now, the blue lettering is Kármán sequences that have been added, and the red letters are the ancient sequences. Now, let's look at the details here. Once again, we see that the Neanderthals all cluster together, but they share a common ancestor with members of the A00 haplogroup. This A00 is from the petrodata, and these two are from the Kármán data. And they have a common ancestor that lived here. And that man shared common ancestry with the A2 haplogroup, who lived here. And that man shared common ancestry with the Denisovans and the A3 haplogroup here. Now, if we put the flood at 2500 BC, according to Jensen's work, the B haplogroups, which are here in the light blue lettering, the B haplogroup split from the A haplogroups about 2100 BC, according to Jensen's analysis. And that would mean that the common ancestor of the Neanderthals and Denisovans with the A haplogroups lived in 1900 BC, which would make Neanderthals contemporary with the patriarchs Abraham, Isaac, and Jacob. I also compared the sequences using a Jacquard index. And a Jacquard index, if you think in terms of Venn diagrams, it's the intersection of the two circles divided by the union of the two circles. So here on the left I have the 5 ancient specimens, and across the top I have sequences from the Kármán data. And in red are the closest matches. And we see that all the ancients are most closely associated with the A1B1A haplogroups. Mesmakia specimen is actually equally close to the A1B1B haplogroups. I also did a principal component analysis using just 2 sequences. Here are 2 ancient sequences, the Mesmakia Neanderthal and the Denisovan 8 Denisovan, because those 2 had the most data. The others were very sparse, so I only used those 2 sequences. And we'll see that they cluster closest to the A haplogroups, who are descended from Ham. The other Ham descendants are the B haplogroups and the E haplogroups. The Shem and Japheth sequences are in the green and the orange here. So we see that by principal component analysis, once again, the Neanderthals cluster closer to the Ham descendants in the A haplogroup. I also reconstructed the phylogenetic tree for Mesmakia 2 specimen descending from Noah. Now, we have Noah here as the root, and these alphanumerics here are single nucleotide markers characterizing this tree. If the marker is surrounded with a red box, that means that the Mesmakia specimen carries the alternate or the derived allele for that marker. So first we have Noah giving rise to Ham, and I have 2 markers for that transition, for that inheritance, and I could have used 7 or 8 other ones, but of the 2, the M522 SNP, the Mesmakia specimen carries the derived allele for that marker. Ham then gives rise to the A and B groups, which split from the E and H groups marked by M186. Now, there was no genotype recorder for this marker in the Mesmakia specimen, so I don't know whether he carried the derived or the ancient allele. The A and B haplogroup split, and this is characterized by M42 and M126. A split into the A1 and A0, characterized by P305 and V221. A1 split into A1B and A1A, characterized by M11015. Now, A1B1A gave rise to Mesmakia, and this is marked by A21560. Now, ISOG, which is the International Society of Genetic Genealogy, calls A21560 a specific Neanderthal marker. And I looked for this marker in the 2,297 Y chromosomes I have available from 3 large online databases, and not a single specimen carried the derived allele for this marker, but it is found in the Mesmakia 2 specimen. The other 2 Neanderthals, the SPY specimen and the El Cidron specimen, had no genotype call at that position, and neither did any of the Denisovans. So let's talk about the unresolved issues. There are problems with the data. One is there's a very high mutation count in the ancient Y chromosomes. The Mesmakia Y chromosome sample has as many mutations on its sequence as the A00 sequences from the modern databases. And this is counterintuitive because if Neanderthals are only 20 to 25 generations removed from Noah, but modern sequences are 150 to 180 mutations removed from Noah, I would expect more mutations to have accumulated on the modern specimens. So why does the ancient Mesmakia's Y chromosome have as many mutations on it as the modern A00 specimens, for example? And I think there's two explanations. One is maybe the Neanderthals had a much higher mutation rate than moderns. And the other possibility is many of the SNPs on the ancient Y chromosome may be artifacts of decay rather than actual inherited mutations. Another problem was absent genotype calls. In the 6.9 million bases of sequence in the database from the Max Planck data, there were 88,248 variable positions. And of these, in the moderns, 98% of these positions had genotype calls, but for the ancients, only 35% of these positions had genotype calls. The Mesmakia specimen had about 85% genotype calls. And the final problem is low coverage sequencing. Coverage has to do with how many times the same sequence of DNA is resequenced to be sure the base calls are correct. Now, these are the coverages for the 5 ancient specimens, and we see that the Mesmakia sample had the highest coverage at about 14 on the average, and it went from about 8 to about 18-fold coverage. The SPY specimen, however, had only about a once-over coverage, so we see there's a great variability in the amount of coverage in these 5 samples. And this brings the data into question. Are these results trustworthy? What would the average physicist think about data like this? Small sample size, low coverage, decayed DNA, data anomalies. I think Einstein would be quite skeptical. So let's talk about the assumptions. Neanderthal remains are post-flood. That's an assumption. Number 2, I assume that the timescale is 4,500 years since the flood. Now, these first 2 assumptions I don't think are controversial among young Earth creationists. The third assumption is, however, that the SNPs are inherited mutations rather than artifacts of decay. So what can we say about Neanderthal? Who is Neanderthal? I think these 4 points are well established by this analysis. 1, Neanderthal is fully human. Let's get over the idea that he couldn't speak and that he couldn't make tools and all. He was fully human according to the material culture and the evidence of the paleontological studies. Secondly, Neanderthal is descended from Noah in the lineage of Ham, and he is traveling as a branch of the A haplogroups, according to this analysis. And this means that he's a contemporary of the biblical patriarchs. Now, there's still some major questions about Neanderthal. One is, why no Neanderthal remains in sub-Saharan Africa? Neanderthal, if he's in the A haplogroups, most of the A haplogroups today, almost all of them are in sub-Saharan Africa. Now, Neanderthal remains were found in sub-Saharan Africa, but they reclassified them as Homo heidelbergensis remains, and some of them as Homo erectus remains. And so that's how Neanderthals are no longer found in sub-Saharan Africa. And so this is a potential problem. Another possibility, however, is that Neanderthals split from their cohorts in the A haplogroups soon after the Babel event, and the Neanderthals traveled into Europe, whereas the other members of the A haplogroups traveled into sub-Saharan Africa. Another major question is, and I find this really intriguing, is Neanderthal actually an extinct lineage? Everybody says so, and they've always said so. But there are several things that make you think they might not be extinct. Number 1, the flood was only about 150 to 180 generations ago, and Neanderthals are post-flood. Secondly, there is Neanderthal DNA in every person in the world today. And thirdly, it just hasn't been that long, and we haven't looked hard enough to find Neanderthal sequences. So I think we need some help on this question. Is Neanderthal actually an extinct lineage? And so I'm going to get help from the Iceman. Let's call in the Iceman. Frozen Fritz was discovered in the 1990s at the border between Italy and Switzerland. As the glacier was thawing, they found this body coming out of the ice. And at first they thought it was a homicide case. So the Italian police said it's on the Swiss side of the border, and the Swiss police said it was on the Italian side of the border. But then they found out it was a mummified Ice Age man of international interest. And the Italians said it's on our side of the border, and the Swiss said, no, it's on our side of the border. But the Italians won the argument, so the specimen is in the Tyrolean Museum in northern Italy. Now, poor Otzi the Iceman, they call him Otzi, he is the most studied mummy in the history of mummydom. They went over him with a fine-tooth comb, every scar, every tattoo. He had a lot of tattoos. They examined all the implements and clothing that were with him. They even sequenced the blueberries in the little pouch he had with him. His axe is a copperhead axe and was hafted onto a wood stick. It had 3 human bloods on the axe blade, which means he was either in a battle before he died or he was some kind of axe murderer. We just don't know. Now, they CT scanned Otzi from head to toe, and they found that he died of a thoracic arrow wound. There's an arrowhead lodged under his left subclavian, and it severed the left subclavian artery. And I think the way he's lying here, I think he was trying to staunch the blood by lying on his side with his arm drawn across. They also sequenced his DNA, and they found that the Y chromosome was in the G haplogroup, G2A4 haplogroup. And they looked around the Tyrolean Alps, and they found 15 men in Otzi's lineage, either direct descendants of Otzi or a close relative of Otzi. So his lineage is not extinct. Now, this raises the possibility for Neanderthal, because ISOG has said that S&P A21560 is a distinct Neanderthal marker, and this is a G to C transversion. So I'm going to make a prediction, and they say young Earth creationists don't make predictions, but I'm going to make one, that if we continue to look around Europe, we may find a few individuals in the A haplogroup who actually carry this marker are in the Neanderthal lineage, and the lineage is not yet extinct. Now, I may be wrong on this, but whether I'm right or wrong, it's still the case that the Word of God is true, and it's explaining what's happened to us when it says that from Babel did the Lord scatter them abroad upon the face of all the earth. Genesis 11:9. Thank you for listening.